And I think we'll take a shorter time today. We'll not take a long time. Probably within an hour, we should be done. So we'll be learning about viral isolation and cultures. So basically, cultivation of viruses. So as we had learned before in the viral diagnosis class, the purposes of isolation of viruses is to identify viruses in a clinical specimen. For example, if you want to diagnose a viral infection from a clinical specimen, you can use viral cultures to identify that particular virus, or to prepare viruses for vaccines. So this is more than diagnosis. It is more applicable in viral cultivation than diagnosis, because for diagnosis, there are newer methods that are, the turnaround time is shorter, and they are being employed more than cultivation of viruses. So nowadays, cultivation of viruses is more applicable for preparing viruses for vaccines, or to just do detailed research on viral structure, its multiplication cycles, and other effects on the host cells, such as to study the pathogenesis of that viral infection, or to even study the immune response of that virus. So it's not routinely done, as I have said. It's mostly done in research organization on research institutes. So we all know by now that viruses cannot be grown on artificial culture, because viruses are obligate intracellular microorganisms. They need cells to survive. They need to take over the machinery of a cell so that they can replicate and, infect other cells. So they can be cultivated on embryonated eggs, as we saw before. We can use a tissue culture, or it can be animal inoculation can be done. And as I had said, it's not done routinely, because they are more efficient and more newer methods that are used for diagnosis. So these are just used for, most of the time, research purposes. So if we look at the egg inoculation technique, an embryonated egg is usually used. So this is an egg that is around 7 to 10 days. And it's preferred because an embryonated egg will give 4 sites, or different sites, for inoculation of the virus and for cultivation of the virus. And also the egg is immunologically inactive, so it's very suitable, and it's sterile, so, and it's easy to get, other than other types of cultures. So this diagram shows the different sites that can be used to inoculate the virus and cultivate it. So the yellow section shows the yolk sac, and then the section surrounding the embryo is the amniotic cavity. And then the section after the amniotic cavity is the allantoic cavity. And then now the allantoic membrane. So the allantoic cavity is bigger compared to those other different sites, and different viruses are inoculated and cultivated at different sites of the egg, as we shall see on the next slide. So for example, the chorioallantoic membrane is used to isolate and cultivate herpes viruses, smallpox virus, and Rous sarcoma virus, while the amniotic cavity is used to isolate and cultivate mumps virus and influenza. And the yolk sac is for isolating arboviruses such as Japanese bee encephalitis and West Nile virus. But the allantoic site, sac, we saw earlier, it's a little bit bigger, and it gives a better yield for vaccines. Hence, it's used to make vaccines for influenza, yellow fever, and rabies vaccines. So for some of us who may be interested on how this is ideally done, how the inoculation is done. So the egg with the embryo is scanned and or screened for the presence of an embryo first. And then someone who's an expert in inoculating the various viruses onto the different sites does the inoculation by drilling a hole, and then the inoculation is done on the specific site. And then this particular hole is sealed using paraffin wax. And then the egg is incubated in a sterile cabinet at 36 degrees Celsius for about 9 days. And then after 9 days, it is harvested. And once it is harvested, detection of viral growth is based on death of the embryo or defects in the embryonic development, or you can see damages in the membrane. For example, the chorioallantoic membrane, you will see with the smallpox virus, you will see pox formation. And also the embryonic tissue or fluid can be viewed under the electron microscope to visualize the morphology and structure of the virus that is being studied. So this slide just shows visible lesions of pox on the chorioallantoic membrane. You can see the white pox formation that forms with herpes viruses and smallpox viruses. So what are some of the advantages and disadvantages of using the embryonated egg? So the advantages includes it provides a sterile environment for inoculation and cultivation of viruses. It is immunologically inactive, so it's good for studying and cultivating the virus. It's easily available because the hen's egg is easily available to find other than other types of cultures. And several sites for cultivation. We've seen it has 4 sites, so you can use the 4 different sites for isolation and cultivation of the different viruses. So on to the disadvantages, we need a lot of expertise to inoculate the virus on the specific sites. Not anyone can do it. And there is, for those people who are allergic to eggs, we've seen the allantoic cavities used to make the different vaccines such as influenza. These people, this type of vaccines will not be suitable for them. And not all viruses can be cultivated by this method. And definitely there is less yield because the sites are smaller and the egg is smaller. So if you imagine if you need to make a vaccine for so many people, how many eggs would you use? So that's why it gives less yield. So on to now animal inoculation. This is also not routinely done. It's rarely done because basically due to ethical considerations. Some people view it a little bit inhumane to use animals by injecting them with viruses and then, harvesting maybe the organs and killing them at the end of it. So it brings up a lot of legal and ethical issues surrounding using animals. So some of the animals that are used are live mice, rats, rabbits, guinea pigs, hamster, chickens, and monkey. And the viral preparation is usually injected into their brain or blood or the muscle or body cavity, depending on the area of interest. And this is suitable for studying pathogenesis and immune response because you can easily see the immune response. For example, when you infect, let's say, a monkey with a certain virus, and you can study also the pathogenesis of where the virus is is developing and replicating and which other sites it's infecting. So that can be studied using animals. So it's only used if it's the only method that can be used to isolate that particular virus because of, you know, the ethical issues surrounding use of animals. So this diagram shows a little bit of what happens. For example, for the inoculation of the HSV, they can inoculate the virus on the brain of the mice, and then either the mice will die based on the acting of the virus, or it can survive, but then the virus will be latently form latency at the trigeminal ganglia. And once the infectious HSV is recovered, then the virus, the mice can now be sacrificed, and I think they call it euthanized. Yeah, they can kill the rat to harvest the different organs of interest and do further studies on the virus of interest. So this also brings about the ethical issues again. That's why animals are rarely used, and we'll see that that on the advantages and disadvantages, where for advantages, yeah, we've seen that it can be used to study viral pathogenesis and oncogenesis and even immune response. But on the disadvantages, there is also the ethical issues that I've been talking about. And it's also expensive and difficult to maintain. For example, if you need to use animals for your research and your experiments, you have to breed them in large numbers, you have to feed them, you have to maintain them in an environment where they're not getting infections from other viruses and other bacteria, so they should be infection-free, and that makes it expensive. It's also difficult to choose which particular animal to use for a particular virus. So that's why tissue cultures are preferred, and tissue cultures can either be a cell culture, organ culture, or explant culture. So cell cultures are the gold standard. Organ cultures that include use of tracheal ring for isolation of coronaviruses and use of explant culture for isolation of, for example, using the adenoid explants to isolate adenoviruses. These two methods are nowadays obsolete. People never use organ culture and explant culture nowadays. So the gold standard nowadays is the cell culture. And there are three different kinds of cell lines. There's a primary cell line, the semi-continuous, and the continuous cell line, and they are different in that primary cell lines are cell lines from normal animal or human cells. And the primary cell lines, they undergo about 5 to 10 cell divisions, and they can be used to make vaccines or just isolate viruses. And an example of the primary cell line is the monkey kidney cell line for cultivation of adenovirus. So the semi-continuous cell line is basically a subculture of a primary cell line. So their cell divisions now becomes a little bit higher, 50 to 100, and they can also be used to make vaccines. And an example of those are the human fibroblast cell line for cultivation of the cytomegalovirus. The continuous cell line divides indefinitely. There are cells that are tumors, and they are immortalized by using either oncogenic viruses or using chemicals. And an example is the HEP2 that is used for isolation of HSV and adenoviruses, and the HeLa cell line that is used for cultivation of HSV. So HeLa cell line in full is just human carcinoma of cervix cell line, and it has an history. HeLa cell lines were named after a woman called Henrietta Lacks. And this woman had cervical cancer. And so these cell lines were gotten from the cervix of this particular woman, and they've been used up to now for cultivation of different viruses. So for cultivation of the viruses, some of the mediums that are used, we can use the Petri dish or just a flask, and inside you put the viral growth medium that contains the nutrients that are required for the growth of the virus. And also you have to put in some antibiotics and some antifungals to minimize the growth of bacteria and fungi, and the pH of the growth medium, which should also be balanced. So then now the cells will just grow. For example, the primary cells will grow exhibiting contact inhibition, because normally the cells in the primary cell line, they don't get contact with each other. So once you, for example, if your tissue that you're generating the cells from is the lung tissue, the lung tissue is, you mince the tissue, and you can use an enzyme like trypsin to isolate the cells to be individual cells. Then you grow them on maybe the Petri dish like this one here. And when the cells are dividing, they divide and grow as a confluent monolayer, and they don't touch each other. They have contact inhibition. Then for you to make now the secondary cell culture, which is the semi-continuous cell line, you now subculture a section of these primary cells into another Petri dish to make the semi-continuous cell line. But for the continuous cell lines, you have to treat the cells using either an oncogenic virus, or you can use a chemical to immortalize the cells so that they can now continue growing indefinitely and form microtumors as this one. So continuous cell lines are basically, they are not used to, they are barely used for vaccine preparation or for isolating the virus. Most of the time, they are used to generate antigens that will be used for diagnosis commercially in large numbers. So it's only the primary cell lines and the semi-continuous cell lines that can be used to either isolate the virus or make vaccines. So some of these methods we saw last time, so we won't go through them, of recognizing if there is a viral growth in a culture, and this includes formation of cytopathic effect, if you can still remember what interference is, hemadsorption, and molecular techniques like PCR, electron microscopy, and the list goes on and on. So you can refer back to the notes on lab diagnosis to read further on the same. And this slide shows some cytopathic effects from some viruses that are well known to cause CPE. For example, the adenovirus that causes rounding and grape-like clusters. We can see this one down here. And for herpes virus that cause cytoplasmic stranding and nuclear body inclusions. So this gives a list of some viruses that can be isolated by culture, and some that are less isolated by culture, like rubella and measles. And there are viruses that they're uncultivable. They cannot be cultured, such as hepatitis B and HPV also. So you can go through the list to have a glimpse of those. And what are some of the advantages of using cell cultures? Cell cultures are very sensitive and specific. And since you're growing the virus, you have so many viral copies, you can use those for phenotyping and drug susceptibility. Unlike other techniques like PCR, where you don't have the live virus to probably do phenotyping or drug susceptibility. And they're also used for identification of those viruses that are not yet known, that are unrecognized. So the challenges with cell cultures is like it takes long to get the result. And therefore, if you need a short turnaround time, it might not be the convenient method. So that's why they're mostly used for research purposes. And they're susceptible to bacterial contamination that can inhibit or cause problems for viral growth, susceptible to toxic substances that may be present. If any toxic substance that can inhibit viral growth will be present in the specimen, the virus will not grow. And many viruses will not grow in cell cultures. As I said before, viruses like hepatitis B and HPV will not grow. And there are also safety concerns, especially for the workers, those people who handle these viruses. And also, you never know, sometimes someone can be malicious with these microorganisms and cause some biological warfare somewhere. So it raises safety concern issues. So for newer cell culture formats, we have the shell vial culture, and these are the current line that is used is the MRC5 that is derived from the human fibroblast cells. Advantages of using this is that it is rapid, that is, it takes 1 to 2 days. And the disadvantage is that unlike the other cultures that we've been talking about, this type is not, isolate is not available for phenotyping, so that you cannot do drug-resistant testing if you use this type of isolate. And just to go through how this is technically done. So you take an inoculate from the specimen and put it inside the shell vial. So the shell vial contains a monolayer of the cells, of the cultured cells. So then you centrifuge under low speed to give time for the virus that is potentially in the specimen to infect the cells. And then this is incubated at 35 degrees for about 1 to 5 days, 5 days, depending on the type of virus, because some viruses like CMV will take up to 5 days. Other viruses will take shorter time. And then once this viruses has infected the monolayer cells, then you stain it with an antiviral fluorescently marked or tagged monoclonal antibodies. And then on the cover slide, you now view this using a fluorescent microscope. So then the presence of fluorescence under the microscope is an indicative of the presence of the virus in that particular specimen. So another example of the newer formats for cell cultures is the engineered cell lines, and these are most widely used for research for HCV and for HIV. And this involves transfecting a viral receptor into a cell line to enhance susceptibility to the virus. So for example, if you have a cell line that you'd want your virus of interest to infect, but this cell line is not expressing a viral receptor that this virus would use to infect this particular cell line. So you can introduce this virus receptor by transfecting the cells. And most of the time we use recombinant plasmids to put the gene of interest into that particular cell, so that the gene that is now encoding our protein of interest, which in this case is the viral receptor, is expressed in those cells. So once it is expressed, then the virus is able to infect that particular cell line. So with downstream processes, you can select for the cells that have been transfected, and they have means of selecting or screening for these particular cells, because normally when you're transfecting these cells, you're aiming to disrupt a certain gene on the cell also. So based on that gene not being expressed, then you're able to select those particular type of cells, because from that cell line. And then the downstream processes involves now expansion and evaluation, meaning just producing your cells of interest in many copies that you can use to cultivate now your virus of interest. So cell lines are also, insect cell lines are also newer formats, and they are used for isolation of arboviruses such as dengue, chikungunya, and Japanese encephalitis viruses. So an example of insect cell lines include the Aedes albopictus C636 and the Culex tritanurhinchas. This is for cultivation of the Japanese encephalitis virus, and the Aedes albopictus is for cultivation of dengue and chikungunya viruses. So if you're interested to know more about insect cell lines, I'll put a link to an article that describes well the insect cell lines. You can read about that. And these are some of the sources that you can use that can be helpful. There is a lunch review of medical microbiology in the field. Someone asked about a textbook last time. It was the Field Virology 6th edition. You can check it out. I think there's a PDF version online. Even the review, there's a PDF version online, so you can check it out. So I think I'll go ahead and tackle the questions if there are any. The presentation today was that short. So Gabby is asking, the embryo is for so we saw from the next slides how you detect whether the virus has grown or not. So the embryo is used when the embryo dies, then you can ideally tell that that the virus has caused, death, or the virus has caused some form of pathogenesis to the embryo, and hence it has died. So it's a way of telling that there's viral growth in this particular embryonated egg. I can see someone has said chicken. I think I got the question wrong, but yeah, it's a chicken embryo. Hi, Doc, can the viruses be inoculated in the stem cell culture of the particular animal instead of euthanizing the whole animal? It depends on the interest. For example, on the interest. For example, if you are studying about the pathogenesis of HSV, then your region of interest is, the CNS or the brain, yeah? So that is, you want to see the pathogenesis of HSV in that particular organ. So it will be more, easier to inject it on the region of interest and study the pathogenesis of the virus, rather than doing stem cell culture. And I'm thinking stem cell culture is also a little bit, difficult to do, and you need to have the facilities and, the technical know-how in place to do it. And I don't think there are so many people, maybe in our region, that have that capability to do stem cell cultures. What are the ideal tissue or cell cultures for monkeypox virus and chikungunya virus? So for chikungunya virus, I just showed the insect cell lines. It's perfect because the insect cell lines from Aedes albopictus, it's the perfect for chikungunya because chikungunya is an arbovirus. And then for monkeypox, I would say the chorioallantoic membrane, because the pox viruses will show pox formation, and then you can be able to tell, to detect viral growth by that. But again, as I indicated later on the slides, that for cultures, there are some other downstream identification techniques, such as looking at CPE, cytopathic effects, or if it is, let's say, a virus that has hemagglutinin on its surface, you can use hemagglutinin inhibition assay or hemagglutinin assay to further detect. There are other mechanisms like further, you can use electron microscopy. So it's not only the culture, you do downstream tests to be more specific. Henrietta Lacks, yes, Henrietta Lacks. So it's a very interesting story. You can go ahead and read about it. Apparently, this doctor who was treating this lady for cervical cancer didn't even get consent to use those cells, those cervical cancer cells for research and whatnot. But you wonder, was it for a good cause? Because maybe if consent was asked, we wouldn't be where we are right now. So I don't know if it was good to not consent or not, but it's an interesting story. You can read about it. Yeah, even the husband didn't know. It's only 10 years later that they realized that, the cells of the wife are somewhere, doing some magical science. You all should read about her. It's crazy. Yeah. It's a crazy story. Her cells are alive and she's dead. All right. What is the significance of the secondary culture? Like, is it a must for the secondary culture to be prepared? So as you have seen, these viruses can be very, very specific in the types of cells that they'd want to grow in. And since the primary culture is just giving a low yield, the secondary culture would come in to benefit in that it can give a better yield. Because let's say for vaccine preparation, you need a better yield other than a cell line that is just undergoing 5 to 10 cell divisions. And you can't use continuous cell lines for vaccines. So in this case, the secondary cell line will give a better yield than a primary cell line. Please repeat the disadvantage of shell vial culture. So the disadvantage of shell vial culture is that you see, like with the other cultures, you get the live virus, you isolate the live virus, and you can study its phenotypic characteristics. And the phenotypic characteristics of virus that someone would want to study of interest are its drug susceptibility mechanism. Is it susceptible to certain antivirals or not? But in this case, you are not able to isolate the virus in itself, although it gives a a lower turnaround time for diagnosis. It's mostly used for diagnosis. It gives a lower turnaround time, but you cannot isolate this particular virus and cultivate it for studying its phenotypic characteristics. So that is its disadvantage. I hope it's clear. Okay. Okay. All right. Do we say transfection instead of infection as it's happening outside the main host? So this is more of molecular biology because in molecular biology, when you introduce a gene to a certain cell and the gene wasn't there in that particular cell, you say you're transfecting. So microorganisms infect people, but when you're introducing a gene of interest so that it's expressed in a certain cell, then we say transfecting. All right. I don't know what Adang is saying. Maybe he can put his question in an understandable way. Okay, what is the essence of introducing receptors to impermissible cell lines so as to infect them, and normally they are not infected by the virus? So remember, in this case, we are trying to cultivate a virus. And why are we cultivating the virus? As I said before, we want to study the characteristics of this virus, or maybe we want to make vaccines using viral antigens. So we need this virus in so many copies. That's why you have a cell line that is normally not infected by this virus, but you want this virus to infect this cell line so that this cell line can be able to produce for you more copies of the virus. So you make this cell line infect, if there's a word like that, infectable, by introducing that viral receptor so that now they can be infected and then you can produce many copies of the virus. Please explain culturing of hepatitis A virus, because ideally its culturing is possible but not commonly done due to lack of efficient tissue systems. Which tissues are possible for HAV culturing? So as we have said on the disadvantages of culturing is that not all viruses will grow, and even if they grow, they might not grow efficiently to produce products that you can use for your study of interest. Let's say if you want to make a vaccine or if you want to study the virus. So for hepatitis A, I know its culturing is not commonly done. For the efficient tissue to culture the system, I may need to confirm that. So please remind me next week on Wednesday so that we talk about this before we start our class on prions, because I have a class with you on Wednesday. But just know that HAV is not commonly, its culture is not commonly done. So for those type of viruses, other techniques now become more suitable, especially for diagnosis, because diagnosis techniques are currently so many, there's serology, PCR is there, and they're very efficient. But now for its research and studies, then there is where now there is a problem. Human fibroblast cell line is used to culture which virus? So someone has answered you there. Okay, the GAN is for cytomegalovirus. Okay. So topic is, we are at this point and someone doesn't know the topic. All right, so the topic is viral isolation and culture. It's basically a part of what we had done on lab diagnosis previously. So it's a repetition of what we did. So I hope it's more understandable now. If cancer cells and human cells manage to integrate in harmony, will we achieve immortality? If cancer cells and human cells manage to integrate in harmony, I don't know where this, in which context this question is coming from. So I don't know whether it's coming from an aspect of virology or an aspect of just normal, you know. Is it really, do you want it answered in virological aspects or other aspects? Because it doesn't sound like virology. Please let me know, Bunyaviride. Please go over insect cell line once more. All right, I'll go through that. Okay. After cell culturing, then what do we use the culture for? So I've said from the first slide, the purpose of cell culturing can be diagnosis, which is rarely done from the specimen of interest. But the two main reasons as to why someone will do a cell culture to cultivate a virus is to either make a vaccine. So it's basically for research purposes, make vaccine, or two, to study the virus itself, study its replication technique, study its cycles, or study its pathogenesis, study its immune response. So all those are there. So it's more of research and less of diagnostic. So Kamunya says, don't think so. Human cell growth, although limited, is well regulated. Cancer cells just replicate continuously bila adabu. So I think Kamunya is answering. Oh, there was someone up here who was talking about cancer cells and human cells. I didn't understand whether it's from a context of virology or a context of just general knowledge, but tell me if Kamunya's answer is not satisfactory. Technically, it is, you need to upset a cell's growth regulating kidogo to make it cancerous. All right, so someone asked about insect cell lines. So I'll put up the slide and then we'll go through it again. So as I said before, this is one of the new cell culturing formats, and it is mainly used to isolate arboviruses like dengue, chikungunya, and Japanese encephalitis. So I gave two examples here of the insect cell lines. There is the cell line for Aedes albopictus, which is used to isolate and culture dengue and chikungunya. And then there is the cell line for Culex tritanohincus that is used to isolate and cultivate Japanese encephalitis virus. I hope that's now clear. And if you want to read more, there is a link to the article there. I hope it's now clear. So can the cultured viruses be stored at an inactivation temperature the way bacteria cultures can be preserved? So an example is the shell vials. So the shell vials are manufactured. They're just manufactured and you buy them. You buy the cell cultures that are already prepared. So before you use them, you store them at -70 for the longest period of time that you can think of. When you want to use it, you take it out and then use it to isolate and cultivate your virus. But of course, once you've used that particular shell vial, you cannot use it again. So I hope that answers your question, Kefa. Any other question? Any other question on virus isolation and cultivation? We are almost, I think we went a little bit too fast. I hope we understood something. Let's make use of the books that I've put as a source there. I find some of those books easier to understand some of these techniques that might sound Greek, but read through those, they will be helpful. So why are tracheal rings used for to culture coronaviruses? So it all goes back to where does the coronavirus infect? Yeah, and where would it, on which cells would it grow better? So that's the question. So you know the infection sites for the coronavirus, right? So then yes, so that is, this S2 is the receptor. So the cells on the tracheal rings, or on the, yeah, or basically the cells that are expressing the S2 receptor will be, the coronavirus will infect those cells. So and we all know the route of entry for coronaviruses, it's through the oropharynx, right? So then that's why the tracheal rings become the organ of choice for the coronaviruses. So it's S2 receptor, Benson, yeah, not SE2, it's S2 receptor. So the cells that will be expressing S2 on the tracheal rings, they'll be cells of choice for the coronavirus. But remember, this is now an organ culture, and it's not a cell culture. Okay. I hope that makes sense. All right. Any other question before we go and enjoy our Friday? All right, team. So I think let's call it a day. I'll send the slides to Kelsey, and let's meet on Wednesday. Our class on Wednesday will be on prions. So I'll see you then. Otherwise, enjoy your weekend.
Thank you.
Thank you.